Good afternoon, everybody. Welcome to the NARI Seminar series. My name is Frances Batchelor and I'm the Senior Principal Research Fellow and Associate Professor here at the National Ageing Research Institute, and very happy to see you all online. I'd like to start by acknowledging the traditional owners of the lands on which we are meeting today. And for me, that's the Wurundjeri Woiwurrung people. I'd also like to acknowledge the traditional custodians of the lands, waters and skies across Australia. And I pay my respects to elders past and present and to all Aboriginal and Torres Strait Islander peoples. Firstly, a little housekeeping regarding Teams. The chat function has been turned off. However, at the conclusion of the talk, we will have some time for Q&A. And if you have questions during the talk or at the end, you can submit them through the seminar using the Q&A function, which can be opened using the tab at the top of your screen. Today's seminar will be recorded and uploaded to our website within a few days. Please keep this in mind. If you turn on your audio and video to ask a question, you will be included in this recording. So I'm really pleased today to say that we will be hearing from Professor Séverine Lamon. Professor Lamon is Professor and Faculty of Health's Associate Dean for Research at Deakin University, where she leads a laboratory focused on skeletal muscle physiology, ageing and sex differences. Her work integrates human cohorts with cutting-edge molecular and multi-omics approaches to understand the biological drivers of musculoskeletal ageing. She's established one of the most deeply phenotyped male and female muscle ageing cohorts internationally, providing novel insights into how ageing trajectories differ between sexes. Beyond her research, she plays an active leadership role in advancing sex and gender inclusive science and contributes to national policy and strategy in health and medical research. So we're very much looking forward to your presentation today, Séverine.
Thanks so much, Frances, and thanks so much for pronouncing my first name properly. That's pretty rare. So welcome everyone, and thank you for attending this lunchtime seminar. I'm really pleased to be there. So as Frances mentioned, I specialize in skeletal muscle physiology with an interest in sex differences during the aging process. So today I have been asked to present about one aspect of sex differences, which is the female aspect and how female sex hormones interact with skeletal muscle during the aging process. So speaking to an aging audience, I do not have to remind you how aging will become more prevalent over the years and how the world population age 60 years and older will only become more prominent in the next 5 to 25 years. So skeletal muscle, for those of you not versed in skeletal muscle physiology, is the largest human tissue. It makes up about 40% of total body mass, and it is made of 80% of proteins. It is the functional basis for our posture, locomotion, but also for our capacity to exert movements. And it is also an essential tissue to whole body metabolism, nutrient storage, and thermoregulation. And for all of these reasons, maintaining the quality of our muscle and its structure, its mass, and its function as we age is an essential component of healthy aging. So as I said, during aging, skeletal muscles play a vital role, and it's really vital because if you don't have it, you basically die, in keeping posture and capacity for movement and metabolism, and critical component of healthy aging overall. However, muscle aging is a pervasive process that presents as a progressive series of both visible and invisible dysfunctions That ends up resulting in severe functional impairments, including things that you can see in everyday life. If you look at your grandparents or elderly people, reduction in strength, reduction in mobility, reduction in balance, and reduction in endurance capacity. And these declines directly result in decreased quality of life, increased mortality, and increased morbidity. So basically, this is what happens when our muscle ages. We usually reach our peak muscle mass, function, and performance around 25 to 30 years of age. And then during adult life, to an extent, and also depending on the amount of physical activity that we do, we can maintain this peak for a little bit more. And then in older life, no matter what we do, we will see a functional decline in our skeletal muscle function. And this is where exercise becomes really important, because it can help minimizing this loss and keeping the elderly people above this level of low physical performance, more importantly, above this threshold of disability that you can see on the graph. So the first thing that happens when the muscle ages is a functional decline. This is accompanied by a structural decline. So on the left-hand side, you can see this is a healthy muscle of a 20-something-year-old, and the dark part is actually the meat. It's the muscle itself, the bone in the middle, and a very low amount of fat around it. If you look at the right-hand side, you can see what the muscle looks like in an 80-year-old. The tissue is marbled by fat infiltration. More importantly, it is also much smaller, and its composition, it's different as well, as I will show you in a minute. So the third type of alterations are around the composition of the muscles, and those are invisible molecular alteration. Again, in the young person, you can see this really nicely well-defined muscle fibers that look healthy. Each color represents a different type of muscle fiber. So we have slow fiber, we have fast fibers, we have mixed fibers that allow to perform different types of movements. And on the old side, you can see that these cells tend to become more round-shaped. There's more space between them. They're not as well defined. They are smaller. And also the colors are not as well defined, meaning that they're starting to sort of hybridize with each other. So this is what happens no matter what we do in every individual, male or female, who is aging normally. There's, however, a catch, which is that males and females age very differently. On the muscle level, it presents in many different ways, which I will explain in a minute. But I think the basis for the fact that aging is a very different process in males and females stems from a few more systemic characteristics of males and females. The first one being that females actually live longer, but they are more susceptible to the negative metabolic and functional consequences of age-related muscle decline. And this is for a few reasons. They live longer, so they have a longer lifespan to deal with declining muscle mass. They often have lower muscle mass to start with. They often consume less dietary proteins. If you remember, the muscle is made up 80% of protein, so this is actually really important to maintain muscle structure and mass. They are also less physically active than males, and obviously the loss of ovarian hormone and follicular activity that follows menopause is a big factor, which we will dig into a bit later. So that means a few things, and the most important one being that the aging trajectory of the female skeletal muscle and its functional outcomes do not mirror, and more importantly, cannot be inferred from male-specific knowledge. And this is not surprising, because if we look at skeletal muscle, we can see that male and female skeletal muscle present many differences, not only during aging, but at many stages of life. A few years back, we wrote a review where we started to list these sex-specific phenotypic traits such as muscle growth, muscle regenerative capacity, fiber type distribution, muscle function, and muscle metabolism that were different both at baseline but also in response to exercise. And exercise will keep coming up in my talk because, as far as we know, it remains the most efficient way to maintain appropriate muscle mass, function, and metabolism during aging. And instead of going into the details, I've got a really cool little video which I like showing. So this is at the World Championship in Doha in 2019, and they had a really interesting event, which was a mixed relay. And I think this shows you everything you need to know about functional differences in skeletal muscle between males and females. In this relay, the teams could decide the order of the runners. So they could be, for example, woman, woman, man, man, or woman, man, doesn't matter. But in that case, you can see that this female is running in the middle of a pack of males, and she basically stands 0 chance, despite being probably the fastest in her country. So I like this video because it's very good at explaining what I'm trying to say in terms of sex-specific differences in skeletal muscle at the functional level. Now, if we look at the molecular level, we won't be surprised to find out that at baseline, skeletal muscle has up to 3,000 genes that are differently expressed in males and females. And if you look on the graph here, it appears here. So it is not from a quantitative, but from an effect size perspective. In other words, how much differentially expressed these genes are in terms of effect size. It is actually the second most biased sex tissue in the body, second only to breast tissue. So it's a very highly sex biased organ. And over the years, my team has shown many sex differences in skeletal muscle at the molecular level. So we've shown differences at the methylome and transcriptome level, again, both at rest and with exercise. We've shown differences at the microRNA level. So all of this doesn't come as a surprise, but it had to be done for us to have a baseline to work from. So if we summarize what we've learned so far, there are sex differences everywhere in skeletal muscle at the functional level, where males usually have more strength and greater muscle mass, while females have a better endurance and are more resistant to fatigue. We've seen that there are phenotypical differences in terms of fiber type compositions. There are metabolic differences in terms of mitochondrial content, which explains why, for example, females are better at resisting fatigue, muscle fatigue. There are differences in fat infiltrations. And finally, there are differences at the molecular levels, which influence how the various signaling pathways and gene expression downstream are being activated. And if we look at, but where do these differences come from, which has been main topic or focus of my work over the last 10 years, I think that to address this question, we first need to understand a very basic concept. And this concept is why are males still the default subject in health and medical research. And this is something I wrote 6 or 7 years ago in the conversation, and I was quite proud of this article because the timing was sort of perfect. The discussions surrounding the study of sex and gender differences in science, especially in health and medical research, were emerging. And we started to gather a group of people, of like-minded people together. And I'm not taking direct credit, but some of the work that we've been doing over the last 6 years has led to the implementation of the NHMRC statement regarding sex, gender, sexual orientation, and variations of sex characteristics that has been implemented for every NHMRC-funded grant in January 2026. So that was the start of a 7-year journey which has led to a very concrete policy change at NHMRC level. So while male is still the default in medical research, I'm sure you all have many ideas about why that might be the case. But I would like to make the point that no disease or condition, basically none of them, is sex agnostic. And if you look at this Lancet paper, they've shown, like, they've grouped diseases and conditions within like larger categories, if you like respiratory infections, nutritional deficiencies, or musculoskeletal disorders. And they've mapped them by age on the y-axis and by prevalence in males versus females on the x-axis. And I love this graph because it basically shows you that there is no zero groups of condition that presents at the same prevalence in males and females at any time in age. So this leads me to a paper written by my colleague, an editorial written by my colleague, arguing that neglecting sex and gender in research is actually a public health risk. And I couldn't agree more. So back to the origin of sex differences. So it's quite simple. There are two main aspects of sex differences in humans and mammalians. And the first one are sex chromosomes, and the second one are sex hormones. And this leads to the major sex differences in humans. There are obviously other factors, environmental factors, societal factors, but they're the two biological factors we focus on. Because the research I'm going to present today is focused on human, and this is not all the research I do, but this is the research I have decided to present today, we're going to focus on the one aspect that we can actually control in humans, which is sex hormones. Sex chromosomes, unfortunately, tend to be fixed. Not the case in animals again, but today we're going to focus on sex hormones in humans. So sex hormones are all derived from a common precursor, which is cholesterol. It is converted into a first steroid precursor called pregnenolone, which is not shown on this graph. But a byproduct of pregnenolone is progesterone, which is our first ovarian sex hormone produced mostly in the ovaries. And progesterone can then produce one of several metabolites, and I've here highlighted the two most important ones for our talk. Testosterone, which is the main androgen or the main male sex hormones, which is produced in the male gonads and binds to its own receptor called the androgen receptor. And on the other side, estrogen, which is the main female ovarian hormone, which is also produced in the female gonads and binds to its own estrogen receptors with a few nuances. So today we're going to focus on our ovarian sex hormones, progesterone and estrogen, and their individual receptor, the progesterone and estrogen receptors. So as you know, ovarian sex hormones fluctuate both across the menstrual cycle and across the lifespan. And if you're familiar with the female menstrual cycle, you know that estrogen peaks just prior to ovulation and progesterone peaks during the luteal or the post-ovulation phase. And both return to sort of a baseline level at the end of the menstrual cycle. And they are both produced in the ovaries, but they exert their effects everywhere in the body, including in the brain and in the muscle. So the main function are reproductive, and you can see the little cartoons here showing what their main functions are during the reproductive cycle. But what we need to keep in mind here is that the estrogen and progesterone, and to an extent the androgen receptors as well, are expressed across many tissues of the body, and they can act on many more tissues, not restricted to the reproductive tissues. So across the lifespan, this is a similar story, and you would know that menopause is characterized by a drastic decline in the production of endogenous estrogen and progesterone. And when graphed, it looks like something like this. So that's estrogen, but progesterone behaves pretty much the same way. It increases at puberty in females, and then it sort of plateaus during reproductive years with these menstrual monthly fluctuation that we mentioned before. And then during menopause, there's a drastic decline in the production of the main estrogen called estradiol, which is then replaced by other sorts of estrogens which are not as potent as estradiol. And this means that females end up spending over a third of their life, maybe more, in an ovarian hormone deficient state. So the next question for us is, we know that sex hormones vary across the lifespan and during aging, and we know that sex hormones play an important role in skeletal muscle, as at least we assume it is based on our male data. So what do we actually know about the role of sex hormones in female muscles? So I think there's two things to be aware of before we address this question. And the first one is that both sexes actually produce both male and female sex hormones, just in different concentrations. So estrogens and progesterones are produced at concentrations that are about 5 times higher in females than in male. On the other hand, testosterone is produced at concentrations that are about 10 to 15 times higher in males. This is important because lots of assumptions have been made over the year about the role of testosterone in female skeletal muscle, and I'll briefly touch on that in a minute. The other important point is that the receptors for sex hormones that we've talked about, they are present in the skeletal muscle of both sexes. So you can find the progesterone receptor, the estrogen receptor, and the androgen receptor on the membrane of skeletal muscle cells in both male and female skeletal muscle. So a few years back, that's nearly it was pre-COVID, I think, 6 years ago, we wrote a review about the effect of sex hormones on skeletal muscle adaptation in females. And I'm not asking you to look at this graph, but the literature was scarce. We knew nearly nothing about the role of both testosterone, but also more surprisingly, estrogen and progesterone on female skeletal muscle. There were few studies, often conflicting findings or findings that could not be confirmed by other studies. So we decided, yeah, we have to do our own studies here. And this was probably a good idea, because if we look at mere associations between sex hormones and our muscle outcomes, we can see that they are extremely sex specific. And this is just pilot data from my lab, basically showing the associations between testosterone levels and a range of muscle outcomes, including total body mass, total body fat, 1 RM on the bottom right, which is a representation of muscle strength, bone mineral density on the top left. And without any statistics here, you can see that these associations are strongly sex-specific. And if we do the same with estrogen levels and muscle outcomes, we can see that it's the exact same thing. So these associations with a range of muscle outcomes are extremely sex-specific. And this is one very good reason to conduct female-specific, or at least sex-specific, research on skeletal muscle. Because even though the same hormones are at play and acting on the same receptor, we are working with a highly sex-biased tissue to start with, and clearly the associations that we can find between each of the sex hormones are very sex-specific when we look at their effects or their associations with muscle outcomes. So to start delving into the research, I'll show you a little case study using a study we did back, I think started in 2018, and then it was halted by COVID and was finished after. But anyway, the first thing we looked at was the role of testosterone in female skeletal muscle. And testosterone is interesting because I told you it's the major androgenic, so the major male hormones, which is typically associated with masculinizing traits. And testosterone levels are also, testosterone is found in female circulation, but at levels that are 10 to 30-fold lower than your typical males. And it exerts its action via the androgen receptor, which is expressed at the membrane of the skeletal muscle fiber. And I've been fascinated by the role of testosterone in female skeletal muscle for a range of reasons, and one of them being the whole controversy that took place about 10 years ago around hyperandrogenic women in sports, women who naturally produce abnormally high level of testosterone, being phenotypically females but producing levels of testosterone in the male range, and being disqualified from athletics competition on the basis that testosterone had the same or played the same role on female muscle than it did in male muscles, which we ended up rebutting. And there's very nice mouse data showing that if you knock out the androgen receptor in mouse muscle, you can see that male on the left-hand side lose about 20% of their muscle mass, meaning that if you remove the capacity of testosterone to bind to the muscle and exert its effect, you decrease strength by about 20%. However, if you do that in females and knock out the androgen receptor, there's absolutely no differences in muscle mass and strength, suggesting that androgens may not be necessary to reach peak muscle mass in females. And we ended up doing 2 studies around that, one epidemiological study and one intervention study, to try and debunk this idea that muscle strength, mass, and performance was directly linked to total testosterone levels in females. And we actually did that quite nicely. We had a big cross-sectional study, a large population-based cohort where we demonstrated that total testosterone was not associated with lean mass and markers of muscle strength in premenopausal females. And then we had a nice intervention study that showed that, again, there was no association between testosterone and muscle mass and strength in females. And so we concluded that despite lots of beliefs, total testosterone is not predictive of muscle mass and function in females. Also, we know that it's very predictive of muscle mass and function in males. So that was sort of a launching pad for the research we would do later, which was to try and elucidate the role of sex hormones, especially ovarian sex hormone in female skeletal muscle, and especially in female skeletal muscle aging. And when we conducted the first lit review about the role of estrogen in female skeletal muscle aging, we found that there were few studies, again, inconsistent findings, and that the literature was quite poor overall because low sample sizes, but more importantly, a lack of control to, for example, define menopausal stages properly, or to control for the menstrual cycle, and high risk of biases as a result. So again, you don't need to read everything about this slide, but there were lots of hypotheses around the role of estrogen in female skeletal muscle aging, which may or may not have effects on muscle mass function, damage, and energy metabolism. But a lot of these findings were actually reflecting conflicting evidence or investigation by a very low number of studies. So we decided to establish our own cohort and explore the role of sex hormones in female muscle aging using our cohort study. Before that, we quite liked the approach to start from an epidemiological population-based perspective to really look at the big numbers, and then to move on to our own tightly controlled study in-house. So here we used the Baltimore longitudinal study of aging, and we used this established cohort to investigate both the cross-sectional and longitudinal associations between muscle outcomes and circulating sex hormones. And because of the wealth of data that has been collected in the BLSA, we were able to adjust all our models for important confounding variables, including height, physical activity, comorbidities, ethnicity, and follow-up time. Most importantly, we were able to adjust these models with age, and I'll explain in a minute why this is really important. And we found that after age adjustment, there were very significant associations between estrogens and the main markers of skeletal muscle mass, appendicular lean mass, and relative appendicular lean mass, meaning that the more estrogens remained in elderly females, the more chances they had to maintain muscle mass. And here I'm saying it's really important to pick up on the fact that these models were adjusted for age, because in aging females, age declines, well, age increases at the same time ovarian sex hormone production and concentration decreases. Therefore, it's very hard to tease out the effect of aging alone from the effect of the decline in ovarian sex hormones. And we can only do that with a sufficient sample size that allows us to adjust for the effect of age. And this is what we did here. So you could demonstrate that there was an independent association between sex hormones and these markers. On the other hand, we found no advantage of having more estrogens left in the system on any of our measures of muscle function, whether it was force, torque, or muscle strength. So this was an epidemiological study which came with its own limitation despite providing a large sample size. So we decided that we had to establish our own study. And this is how we came to establish the FAME study and its male counterpart, which is simply called the male study. And we have phenotyped 96 males and 96 females, age 18 to 80. We have collected biological samples, including muscle biopsies, blood samples, and urine samples. We have collected muscle functional outcomes, so various neurophysiology testing, DEXA scans, PQCT scans, and strength and power testing. We have accounted for moderators, including menopausal stages and menstrual cycle, and potential confounders in our analysis, including physical activity, sleep, diet, hormone contraception or menopause treatment, HRT, hormone replacement therapy, which is now called menopause replacement therapy, and obviously the menstrual cycle. And because all of these confounders have the potential to affect our skeletal muscle outcomes, and then we did a whole bunch of cellular analysis and omics on our blood and muscle samples. And out of the FAME study, we have published 2 really good papers, which I believe set up the scene to start exploring and understanding the role of these sex hormones in female muscle aging. And I'll take you through these results next. So the FAME study included 96 participants, which were stratified across the decades and included about half premenopausal, give or take, and half postmenopausal females, with a few in the middle, which were perimenopausal. We control for their menopausal status, which I just explained. We included people we included people both taking hormonal contraception and not taking hormonal contraception, because we decided that would adjust for these parameters in our statistical analysis, and we did the same with menopausal hormone therapy. And the first thing we did was to publish a mapping of muscle mass function and molecular signaling in females aged 18 to 80 and their associations with sex hormone. So we collected a whole range of physiological outcomes, including body composition, muscle size, muscle composition, muscle strength and power, circulating sex hormones, and reproductive health characteristic, which I took you through earlier. And I will next take you through the main physiological outcome and the relationships with sex hormones. So you can see these 12 graphs represent various measures of body composition, muscle size, muscle composition, muscle quality, and muscle strength. And if you look at each of the graph, the red line represents the association between the muscle outcome and age after adjusting for physical activity and protein intake. Again, we identify these two confounders are the most important and influential one because both protein intake and physical activity really have the potential to slow down or blunt this muscle aging process, and we didn't want it to mud our results. And the first thing we observed is that pretty much aging is not good for you. And we found a number. So I just removed the graphs that were not were not statistically significant. And you can see that for most muscle outcomes, we have linear association between age and body composition, muscle morphology, and muscle strength. And that unfortunately muscle strength and muscle mass decrease with age and intramuscular fat increases with age. And this was all expected, but it just hadn't been mapped in a female cohort. So this is our baseline. This is what we're working with. Then we looked at various molecular outcomes, and within these molecular outcomes, we included muscle fiber type, muscle fiber proportion, muscle fiber cross-sectional area, and also various measures of muscle quality, satellite cell numbers, transcriptomics, and proteomics. And again, we looked at these molecular outcomes. And again, I'm just going to remove the graphs that were not statistically significant. Again, the red line on the graph represents association after adjusting for physical activity and protein intake, as I just explained. And we found that there were, again, linear associations between age and muscle fiber characteristics during female aging, where, for example, the ratio between slow and fast fibers increases with age, which is an explanation why strength decreases, because fast fibers are responsible for producing more strength. And the size of the average muscle fiber decreases with age, and the amount of fibrosis using a fibrosis marker within the muscle increases with age. So again, none of this is good, but it is establishing a baseline for our understanding of female muscle aging across the lifespan. We then looked at the role of sex hormones with the limitation in mind that this is firstly a cross-sectional analysis, and also this is not an intervention study, so we can't infer causality out of this data. However, when adjusting for age, and again, all models were adjusted for age, physical activity, and protein intake, and then with some additional adjustment for muscle strength model, this is really important, but because again, we're taking out the effect of age to focus on the associations with purely sex hormones. We found that what we found in our EPI study, that is these strong associations between estrogen and progesterone with markers of body fat in lean mass, that is muscle mass within the body, remained very significant, meaning that independently of age, the more estrogen and progesterone was maintained in the body after menopause, the higher the muscle mass and the lower the body fat was. On the other hand, when we look at the same associations with testosterone and other markers of androgenic hormones, we actually found, again, in line with our previous finding, that testosterone and androgenic hormones had nothing to do and pretty much zero association with any of the muscle markers that we were looking at. So then we looked at our muscle biopsies and gene expression patterns across the lifespan, and we found about 1,500 genes after FDR adjustment that were significantly associated with female aging. We conducted gene set enrichment gene set enrichment. And then we took out the main clusters that were exhibiting particular patterns across age. So, for example, going up across the lifespan, going down across the lifespan, but also some interesting patterns where a number of genes would go up until midlife and then would go down, coinciding with the onset of menopause. So a lot of really interesting findings here, which I have summarized on this slide. So if you look at the various clusters, and let's just look at clusters, the top two, cluster 3 and 4, which are the bigger ones, you can see that, for example, RNA processing, transcription, and DNA regulation keeps increasing with age, while mitochondrial respiration and translation, which is a marker of well-functioning muscle metabolism, keeps decreasing across age. And now everything I've shown you to date is from our FAME study, but obviously we did the exact same thing with our control male cohort. And I'm just going to give you a small teaser here into the genes that were differently expressed between males and females across the lifespan. And we found something really interesting. We found that in the younger age groups, there were lots of genes that were differently expressed between males and females in the muscle of males versus the muscle of females age 18 to about 40. But then this number declined markedly with age. So from a gene level, it's like female muscle and male muscle are getting closer on a molecular level during aging. The second thing we found is that these genes that were differently expressed consistently across the lifespan, and this is not overly surprising, were mostly linked to chromosome Y with fewer orthogonal and X-linked transcript, which makes sense because they are the genes which would be less susceptible to the influence of sex hormones. So in summary, the first part of this study demonstrated that aging in females is associated with declines at the whole body, functional, whole muscle, and cellular level, and that estradiol and progesterone may be positively associated with these muscle health outcomes, especially our muscle mass outcomes, less so our muscle strength outcomes, independently of age. I'm just going to pass that and move on to the second part of this study, where we wanted to understand what was actually happening not only at the muscle strength level, but how is the muscle efficiently not corresponding, but I guess interacting with our brain as we age, because you know that exerting any form of movement comes from a very tightly regulated combination between brain signals and muscle signals. And we were really interested to find out whether the declines we were finding in female muscle aging were mostly driven by a loss of muscle structure and strength, intrinsic strength, or were only also related to a loss in efficient communication between the muscle and the brain. So in the same cohort, we quantified the neuromuscular function of these 96 females and investigated their potential association with sex hormones following menopause. And as I explained, the neuromuscular function is just the ability of the nervous system and the muscles to work together to produce and control movement and force, and basically describes how efficiently the brain and the muscle interact to perform voluntary and involuntary actions. And a neuromuscular assessment included a whole range of measures spanning voluntary contraction, isometric torque, and surface EMG measurement to pick up on the electrical activity of the muscle, plus a basic strength test. And this is me completing that test. So you basically have electrodes all over your muscles and you're pushing at, again, an external load, and there's nothing you can do because basically the electrodes are recording what is happening inside of your muscle. And here we found something really interesting. We found firstly that in contrast to our previous outcomes, there were both linear and nonlinear associations between age and neuromuscular outcomes. And the red shaded region in these various measures of the neuromuscular function highlights a rapid decline or an acceleration in the decline of the outcome. And what it means that while some of these associations were linear with age, some of these outcomes, including the first 5 that we've listed here on the graph, showed a drastic decline, basically fell off a cliff at midlife corresponding to the onset of menopause. And that prompted our next study, which we are starting now, which will exclusively look at this menopausal transition to actually understand this pattern of sort of falling down a cliff past the onset of menopause between 40 and 55-ish. And again, we looked at the predictors of these outcomes, and I'm not going to take you through these slides line by line. However, we found that these outcomes could be statistically explained by age, obviously, and by a range of other factors. And no surprise there, estradiol and progesterone appeared again, both positively influencing association, meaning one more time that the more estradiol and progesterone was kept in the system independently of age, the better the outcomes down the track were in terms of the way the muscle communicates with the brain. So if we summarize this part, we found that there were age-associated declines in various markers of the neuromuscular function, and some of them drastically dropped around the time of menopause. And we also found that estradiol and progesterone may be particularly closely linked to how the neuromuscular function declines over the lifespan in a positive way. So the more remains in the system, the better the neuromuscular function remains. And in our next paper, which is about to be submitted, we actually look for a mechanistical explanation for this age-related decrease in muscle strength. And the first few things that we could tease out, and we're not there yet, so wait for the paper to be published, is that aging may affect muscle power more than muscle strength, and that these declines can only be partly explained by the loss of muscle mass. In other words, the brain is a very important contributor. It's not the muscle alone. And we looked at a marker of denervation called NCAM, and basically we also found that damage in this nerve-muscle connection and ongoing muscle fiber repair as a result of this damage contributes to the loss of muscle mass in older females. So that is for the mechanistic explanation that we are looking for now, and this is going to be our next paper, which I hope will be out soon. So to conclude, now that we know that, how can we practically apply our knowledge of sex hormones to help females maintaining their muscle mass and function in later life? So I'm sure we've seen slides like this before, and I cannot reiterate how important exercise is, because everyone, absolutely everyone, will experience age-related decline in muscle strength, muscle mass, muscle power, and overall physical function. What we found is that in females in particular, there might be non-linear declines around menopause, especially in the markers of the neuromuscular function. So if you like, the dotted line in gray might be the hypothetical you would expect from a male subject. However, the blue line is what we're actually seeing in a female subject. And we found potential protective effects of ovarian sex hormones, especially estradiol and progesterone, all across the lifespan. So these hormones might provide a potential biological support throughout the lifespan, including after menopause. As I said, exercise is key, because while we are all facing age-related skeletal muscle decline, I would argue that it's never too late to exercise. If your physical function, which is directly related to the amount of exercise you've performed over time, is high, and you maintain an active lifestyle, the disease burden would be lower with age. However, it's never too late to try and reshape or increase this trajectory and avoid ending up with both an inactive lifestyle and that is linked to a high disease burden. So the best case scenario is to start early to reap the most benefits of physical activity over time. If this wasn't, for many, many reasons, what you did in your younger years, it's never too late to start exercise, to stay active and stay independent while maintaining the quality, the structure, and the function of your skeletal muscle, and reduce your disease risks across the lifespan. So couple of take-home messages. Females around menopause are particularly vulnerable to sharp decline in muscle mass and function, warranting particular attention and individuals' exercise programming. It's never too late to start exercising. Exercise is better than no exercise at any time in life. And because we found this protective effect of estradiol and progesterone, which, and I can't stress that enough, does not allow to infer causal relationships, it could be suggested that for some females, HRT or MRT, menopausal replacement therapy, might provide an additional bonus when it comes to maintaining muscle health and functional capacity over the lifespan. Thank you very much. So this is my team. Without the team, nothing would have been possible. Without the Australian Research Council and my other funding sources, nothing would have been possible, as always. And if any of you is a female aged 40 to 80 in generally good health, we would love to have you across for our next study. Our next study is called the muscle pause study, and is based on this idea that, do you remember the red shaded area on my slide? We want to understand why muscle health falls down this cliff around the menopausal transition, and we want to understand the role of sex hormones. So therefore, we're now recruiting 125 females aged 40 to 80. So if you have any interest, please drop me a line or scan the QR code. Some references and some podcasts for those who are interested in the lay aspects of things. Thank you very much.
Thank you so much. That was a fantastic overview across so many different studies. We now have some time for Q&A. Remember, the chat function is turned off. If you have questions, please submit them through the Q&A function at the top of your screen, which is the two chat bubbles. Speech bubbles, one with a question mark in it. By default, you are muted and your camera is turned off. If you prefer, you can use the raise your hand function in the controls at the top of the screen. If you want to ask a question directly, we can turn your camera on as well. If you have any questions, please put them in the Q&A now. But while we're waiting for that, I guess, yeah, I found that super interesting how I guess there was a lot of assumptions about, oh, I love that, what we know and we don't know, or what was known up until now. And it seems like in sport, they were basing a lot of their decisions that weren't evidence-based. Is that how you see that?
Oh, that's my favorite topic.
Yeah.
Look, sport is an interesting one because I feel like in many ways it is a thermometer for our society, but often we need responses faster in sport than we need them in society in general. And I think the whole conundrum that resulted from these hyperandrogenic females who were biologically females as defined by their chromosomal makeup, but for various reasons were producing male levels of androgenic hormones, this idea to ban them on the pure assumption that total testosterone was associated with or was determinant of muscle strength and performance was just completely wrong. And now there's been some backtrack, but sporting organizations are not the best to recognize when they have it wrong. It's a highly politicized environment. I try to not to comment too much about it, but I think my main message here would be they need scientists on their medical boards. They can't just try to please the public with what they believe is the right thing to do. They really need scientists to do the work. And the same applies to the whole transgender problematic. There's very little data and a lot of decisions being made which have the potential to be knocked back in future years when the appropriate research is actually done. So that's how I would respond to that.
Yeah, that's great. I mean, that really resonates with the work we do at NARI as well, that decisions need to be evidence-based. And, you know, sometimes there just isn't the data to base a decision on, and that's where we definitely need the research. I think, Dave, there are some questions I will ask. Dave says there's growing interest in the interaction between nutrition, the gut microbiome, and muscle health. Do you see a role for sex hormones in modulating the gut-muscle axis? And is this an area your group is beginning to explore?
So I haven't touched the gut muscle axis personally. Well, I just haven't, and this is not something I've thought about exploring further personally, but there are people working around me, also working with mouse model or rat models of sex and aging, who are interested in what is happening in the gut. And I think some emerging evidence will be available sooner rather than later. So it's not a direct interest of mine, but there are certainly people interested in this field who will start producing results shortly, especially on the animal preclinical side of things.
And another question, you mentioned that protein and exercise were two factors that have the most impact or have a lot of impact. You mentioned increasing exercise over the lifespan. To what extent should menopausal and older females increase dietary protein intake?
That's a really interesting one. Look, I think the advice goes across the board, is valid across the board, not only for menopausal females, because in Australia, females tend to not meet their protein dietary requirement at any point in time. There's another issue, which is that the way Australians eat, they have a strong tendency to have, for example, one big meal at night, and this is when you would have, your 200-gram steak, which from a metabolic perspective is not a very good investment, because what you want is a constant supply of proteins across the day. So especially for postmenopausal females and for older males as well, the recommendation is 90 grams of proteins per day, ideally split into 30 grams intake in a way that maximally synthesize muscle protein metabolism. Now, it is hard. It is actually a lot of protein. And while it's easy to have a true 200-gram steak at night, you can't do that at breakfast and lunch as well. So there are many ways to go around this. Athletes have discovered protein shakes many, many years ago, and that's a good way to do it. Or maybe thinking of increasing the dairy protein intake in the morning and at lunchtime. But I'm not going to say the more the better, but the more consistent the better, especially for older adults.
Okay, got two more questions. Have you looked at women post-salpingo oophorectomy and female sex hormone concentration post-menopause? If so, does this vary, and can this be a protective factor?
So I'm not sure I got the whole question. The first one about hysterectomy.
Oophorectomy.
Oh, okay. No, we haven't. No, we haven't. The second one about postmenopausal ovarian sex hormone concentrations. Was that? Yes. So basically, our multiple lifespan cohorts have accounted for postmenopausal ovarian sex hormone concentrations and have actually demonstrated strong associations when the aging factor were taken out of the equation. So I think we can reasonably argue that there are positive associations between ovarian sex hormones at any time in life, including postmenopause, and markers of muscle strength and muscle health in general.
And Sarah asks, are there any associations between exercise type and skeletal muscle aging in women, especially in the transition to menopause cohort, so that we can advise patients?
Absolutely. So during that transition, well, I would argue that cardio is important across the lifespan. Cardio is always important, cardiovascular fitness, for a number of reasons. However, when it comes to skeletal muscle aging in particular, it really has to have a high resistance training component. And this is only because resistance training is the most efficient way to increase muscle protein synthesis. You remember that 80% of the muscle is made up of proteins, and this is how you maintain muscle mass especially across aging. So for clinicians and allied health professionals, it's often a mix of cardio and resistance training. When there is no scope for a mix for many reasons, time, resources, whatever, it's absolutely resistance training that should prevail, because it is the one that will stop this muscle protein degradation process. And if not allowed to increase muscle mass, in some cases, it can allow to maintain muscle mass across the aging transition.
And with that, I think we will stop questions there, and it's a little prompt for me to change my cardiovascular workout this afternoon to a strength training one. I might hit the gym instead of the running.
Yeah.
So thank you so much, Séverine, for this presentation. Please join me in thanking Séverine for this presentation using your clap reactions, which you can find the little reaction button at the top of the screen. Next week, we invite you, not next week, next month, July the 14th, we have a seminar featuring Dr. Penny McKenzie presenting on ageism in cancer therapy. And so we look forward to seeing you at that seminar. Thank you, everybody. Have a great day.
Thank you so much.